All right, it is 2 o'clock, so we will get started here. Reminder that our test 2, is on Friday, so make sure you're preparing for that. The material that we will cover today won't be on that test. Remember, the test 2 folder tells you on Blackboard what is and what isn't going to be on the test. And so we would have stopped with hemoglobin for our test. So this muscle stuff won't be on our test. So let me just finish setting up here. There we go. So I can answer like general questions about the test. If anyone wants to ask like any general questions about the test, if you have any, feel free to answer now or ask now. I'll get the ones like the ones I'll probably get out of the way. Number of questions, I do not know yet because I have not finalized the test. Equation sheet, I will put up an equation sheet for you. I have no problem with that. So I'll probably get that up on Thursday. And you can print that out. Are you going to have math on this test? Again, I haven't finalized. Well, I wrote a draft of it. Honestly, I wrote a draft on Saturday. I have not looked at it since then. So I completely forgot what my draft is. But there is a possibility there will be math on it. I'm going to review that test probably like on Thursday to finalize it. What else? So again, we'll be doing Proctor-U for our test. It will open Friday at 12 a.m. and close Friday at 11:59 p.m. Right? So you'll have all day to do it, 1 hour in length. It's going to be a mix of multiple choice, short answer, all that good stuff, just like test 1. Will there be any matching? No, probably not. There probably won't be matching on there. Can we use the equation sheet during the test? Yeah, so I'll put up every equation that we've covered in this class, and I'll just put it on a sheet. You can print that sheet out and have it with you. That's fine. At this point, I really care that we know how to use equations and that we can recognize equations. And honestly, if you know the name of equations, because if you forget an equation, if you can look it up and know how to use it, that's way more relevant than straight memorization. It is good to know the equation by memory, but yeah, I can compromise a little bit. Can we practice? Not during class. If you want to practice math questions, let me know. We can do a meeting with that. I just don't want to spend like a ton of class time going over math questions again, because we do have lecture videos already that have those up there. Any other general questions about the test? The equation sheet, I will put that up on Blackboard. You can print it out and you can have it with you on the test. What I will do is I will go through the PowerPoint slides and see what equations we've used and throw those on the equation sheet without any words or explanation on it. I do ask that you don't write on it before the test starts, please. Duration is 60 minutes. You have a full hour to do this. Any other general-ish questions people might want to get out of the way at the very start here? Yeah. If you print out the equation sheet, you still get scratch paper to write on. Again, all I ask is that those be blank, your scratch paper. I have nothing against scratch paper. While I think about it, though, So we're doing Proctor U. And I think I mentioned this when we went through the test 1 review, but I'll mention it again. Some things that will flag Proctor U that I have to go and manually check. So please try and minimize this. Is if you leave the camera. So actually, I'm going to stop sharing here for a sec. If you leave the camera like this, that's super suspicious looking, right? So and you'll get flagged and I'll have to look at it. Also, talking to yourself, they worry that someone else could be in the room with you. And so if you're talking, like, I don't know, they could give you like signs like, oh, the answer is that one. To me, that's not a huge deal, but just try to, I know some people like to talk out the questions and I'm kind of okay with that, but just be aware I have to go and review all those. Yeah, the screen thing was weird. They didn't act, so I saw that like some people had like 467 incidents, which was because the screen wasn't the proper size. And so I got to look at those, but yeah, it's not perfect, but it's what we have for right now. Can we use a calculator? Yes, that's fine. Is there anything to do to prevent the screen maximization flag? I don't know. I can ask because it only happened to like a couple people. So I can't say for certain like what set it off versus what didn't set it off. Yeah, it should be maximized. So I don't know why it was throwing a little fit for some people. Any other general questions? Yeah, just the big thing though, what always got flagged was people who would be like this or like this. Just try to, I understand when you're writing, people want to go like this because right, you've been through school and what do you learn in like lower levels of school? Always cover your answers with your arm to prevent cheating. So it's a natural reflex. So I get that. Let me reshare the screen. All right. So if there are no more like generalist questions, we're going to continue on with our material for test 3. Just as a reminder, we were talking about the muscles. So we have 2 different filaments. We have thick filament and thin filament. Our thick filament is made out of myosin, and our thin filament is made out of actin. So myosin is both globular and it's also a fibrous or fibrous protein. So it has a globular head and then it has a very fibrous tail, as we can see in the picture in the bottom left. And then we have actin. We have 2 forms of actin, one that's globular and one that's a filament. So G-actin and F-actin. Again, these are the same protein, just in different forms, just like T and R hemoglobin is the same protein, just in different forms. For the thin filament, we really care about F-actin. So in muscles, we care about F-actin, not really G-actin all that much. So just as a little review here, we talked about the overall structure of the muscles on Monday. So who can remind us for the Z disc? What type of filaments are we anchoring? And at the M disc, what type of filaments are we anchoring? It's not so much I band and A band. What I'm asking for is thick or thin filaments. That's what the question's getting at. Yep, Z is thin, M is thick. Yep. Z disc is light, M is dark, so C is thin, M is thick. Correct. Yep. So Z is our thin disc and M is our thick disc. So our filaments, rather, filaments. So that is just a little review here to get us started, get those brains working a little bit, get the engines fired up. All right. So we're not quite done with the overall structure of our muscle fibers. There's other proteins associated with this. And we have tropomyosin and troponin. And these are on the thin filaments. So on your F-actin in your muscle, you have two proteins, tropomyosin and troponin, all right? Tropomyosin is a homodimer. Let's break down that word. Homo means same, dimer means two. So it's made out of two peptides, and those peptides are identical. So that's what a homodimer is. And it's called a parallel coiled coil, which just means it forms coils. You have a coil that also forms a coil, almost like an old, those old rotary phones, old telephones that had the cords on them, right? So that was like a coil of coils when they coiled up. And what happens is that tropomyosin forms a cable around actin, around your thin filament. So you have your thin filament right here. And then you have your tropomyosin wrapped around it. I guess I should have done a different color, but too late. Associated with these proteins is another protein called troponin. So troponin, which is that like purple thing, binds to three things. Binds to calcium, binds to actin, and to tropomyosin. So troponin is binding to a bunch of things. Okay, so let me just continue with it. I just forgot what the next slide was. So the way this works is that myosin wants to bind to actin, right? That's how muscle contractions work. Myosin wants to bind to actin. That is how we get our muscle contraction. However, if tropomyosin is present, our myosin cannot bind to actin. So tropomyosin blocks this. So to release tropomyosin, we have to get rid of troponin. So the way that this works, think of troponin as a lock, right? So troponin is a lock. And the key to opening that lock is calcium. So calcium opens that lock. So what happens to make our muscles work is that we send a nerve impulse that releases calcium. That calcium will bind troponin. Once troponin is bound to calcium, it won't bind to actin and tropomyosin anymore. It kind of like falls off. When troponin is gone, tropomyosin leaves the thin filament, right? So troponin's gone, tropomyosin leaves. Once tropomyosin leaves, our myosin can now bind our actin, and now our muscles work. So every time you are moving your muscles, right, this process is going on. And when you don't want to move your muscles, like most of the time you don't want to move your muscles, right, you have your tropomyosin and your troponin bound to actin to prevent any muscle movement. All right. So before we move on to like how muscles actually move, is there any questions about the system I just explained here? And that's why it says regulation, because they are regulating myosin and actin binding to each other. So tropomyosin binds to actin when we're not moving. Basically, tropomyosin's wrapped around actin. Troponin is holding all that together. So the last thing I said, I think, was about regulation, in that tropomyosin and troponin regulate when myosin and actin combined. So when myosin and actin interact, that's when our muscles can move. And so when we don't want our muscles to move, we have tropomyosin and troponin interacting with our thin filament, blocking that interaction. Troponin practically locks tropomyosin in place. That's how I think of it. And so that's why I go with that analogy, because once troponin is removed, tropomyosin can unwind. So kind of, yeah. All right. So let me clear this. Are those shown on the picture of the right? Kind of. So what we're seeing on the picture on the left, yeah, I think you meant left. On the right, what we're seeing is, on the right, we are looking at troponin. Yeah. So on the left, that is a electromagnetic image of tropomyosin and troponin binding, where I have to remember this. It's been a while. I want to say like the blue is like your tropomyosin, if I remember right. That's wrapping around. The red is your actin. And I believe the yellow stuff is like the troponin. So the yellow will be removed, which removes the blue, which allows access to the red. I think that it's been a while since I should have reviewed that picture, but it's been a while since I've looked at that picture in your book, but I believe that's what's going on. How does troponin know to bind, unbind? That has to do with calcium, right? So when it binds calcium, it will go through a overall structural change, just like we talked about with hemoglobin. When hemoglobin binds oxygen, we have this whole system of changes that change the whole structure of that protein, which allows it to bind oxygen better. And now a similar thing is going on with troponin. When calcium binds, it's going to change the way troponin looks. It's going to have what's called a conformational change. And then when it binds calcium, whatever conformational change there is, it doesn't want to bind tropomyosin and actin anymore. However, after a certain amount of time, what will happen is that calcium will leave the binding of troponin. It will just float away. Right? It's all based on probabilities, but after a while that calcium will leave and it will readopt the conformation where it wants to bind actin and tropomyosin. So that's how troponin knows. It's all based on conformation. What conformation am I in? And that is dependent whether I have calcium or not. And really all of that is based on physics. This is all physics. And it all has to do with like what kind of amino acids I'm showing, which will relate to what type of intermolecular interactions I have. Is there a certain calcium threshold? After a while, calcium won't do anything. I'm sure there is. I'm sure you can find measurements out there where like after a certain amount you need a certain amount of calcium release to have this work. I don't know what that is, but I'm sure somebody's done that research. All right. If you have more questions, feel free to ask them, but I'm going to be moving on here. So based on what I just said, I think I have a poll for this. Let me just double check. I should have a poll for this. Based on what I just said, yeah, I don't. Okay, I don't have a poll. Okay, what is the best match to describe troponin C? This is a perfect exam 3 question. What is the best statement that describes our friend troponin, better known as troponin C? Is it a component of the thick filament? Is it a calcium binding protein? Is it a unit of the myofibril? Is it a component of the thin filament? Or is it a chemical energy source for muscle contractions? So what do people think? What is troponin? There's only one answer. This is not multiple answer. This is multiple choice. So calcium binding protein, B. Yeah, that's what it is. It's a calcium binding protein, right? It's not a part of the myofibril. It's not part of the thick filament. It's not part of the thin filament. Remember, thick filament is myosin. Thin filament is actin. Troponin is our regulation unit, and it binds calcium. So out of all those choices, the best way to describe troponin is that it binds calcium, a calcium binding protein. So just a good example of what I may ask you on test 3, an example of a question I might ask you on test 3. All right, let's get to the main event. How do muscles work? And they work via what is called the power stroke, right? So we're going to talk about the molecular mechanism of the power stroke. So we're going to start right here. And we're going to start with myosin bound to actin. So we have a myosin head bound to actin, right? So imagine our troponin and our tropomyosin are already gone. We had calcium that was released. Troponin and tropomyosin fell off. And once that falls off, our myosin, or our thick filament, will bind our actin, which is our thin filament. And what happens now is that ATP will come in and bind. So ATP binds. When ATP binds, the myosin head falls off, no longer binds actin. Right? Now, after it falls off, ATP is hydrolyzed. ATP goes to ADP plus phosphate. And during that hydrolyzation event, the myosin head will move. When ATP goes to ADP, our myosin head shifts position, right? After that event, our myosin head will rebind actin. And it will release phosphate. So it will release the phosphate it cleaved off of ATP. Then, very quickly, we have what's called the power stroke. Where our head shoots back to its original position. And it's attached to actin. And so our actin will move as well. So our thick filament is moving our thin filament. After our myosin is back to its original position, ADP is released, and we're back to square one. All right, so usually when I talk about this, I have a demonstration in class with my arms. So I'm gonna try to do that right now, sitting in my seat without the PowerPoint in front of me. All right, so let me try to do that. Okay, so when we start, we have, let me get in camera. Okay, so we have our myosin heads. and I'm bound. I'm bound to actin right now. So imagine there's like 100 copies of me, all facing the same direction. There, now you can see my hands. And we're grabbing onto a rope right here. This rope is actin. So where my head is, imagine that's just like actin. And so what happens is that ATP comes in. I let go of the rope. I'm no longer binding the rope, right? I use that ATP and I burn a phosphate. So my hands shoot forward, right? And I let go of phosphate. I rebind the rope. I let go of ADP and I pull the rope back, right? That's the power stroke. I'm bound. ATP binds me. I let go. Phosphorylate that ATP, shoot forward, right? Release my phosphate, bind again. Release ADP, move. That is the power stroke. That is how your muscles move on a protein basis. That's why we need ATP to do that power stroke. Hope you all like my interpreted dance of the power stroke. Questions about that mechanism? Don't be surprised when I ask you to describe this mechanism to me on test 3. Hint, hint. It does help when you actually see it work, yes. Yeah. Looking at pictures, one thing, seeing like a movie of it, I find is very beneficial. So what happens before the myosin is bound to actin? Okay, so actin, naked F-actin, will have 2 proteins bound to it, right? So actin will have, again, I got to switch my colors. It'll have tropomyosin, and it will have troponin. And so myosin cannot bind actin. What happens is that calcium binds troponin. Once that happens, troponin falls off. Once troponin falls off, tropomyosin falls off. And we're left with just our actin, which our myosin can bind to. So that's like the very beforehand step. Then once actin is naked, that is, there's no troponin or tropomyosin, myosin will, or there's no troponin or tropomyosin, myosin just binds to it. That's what it does. Unless there's ATP also bound to myosin, it just grabs onto actin. How much detail are you looking for when describing it? Basically, can you explain this to me in words? Right? ATP binds, what happens? Then what happens after that? Well, ATP breaks apart to ADP and phosphate. Okay, what happens then? Myosin head shoots. Basically, if I'm looking at this with no words and then like no words at all, you should be able to tell me exactly what's going on. That's kind of like the level I'm looking for. If you want an example, like I ask this question almost every year, go back to the previous test, you'll see how I ask this. I'm not saying I'm going to ask it that exact way, but I'll be asking questions along the power stroke. So that's how your muscles move. Pretty cool. So here's a good question. When people die after a couple hours, I think it's like 6 or maybe more than that, your muscles become stiff and you can no longer bend that person's muscle. That's called rigor mortis. Based on what we just learned, why is rigor mortis a thing? Here's a good critical thinking question. There's no more ATP generated, so the myosin head is stuck. Isn't it because there's no ATP, so myosin doesn't have the energy to move back? Yeah, exactly. There's no ATP. Everybody who just answered right there got it. Yeah, and then the proteins will degrade, and that'll take some time, but eventually that'll happen. So yeah, so when you die, right, a lot of things will happen, but one thing that will happen to you is that eventually your calcium will just start to leak out of like your neurons, and your troponin and tropomyosin, right, will no longer be bound. And then you have naked actin, and then your myosin heads will just bind onto this actin. And since you're no longer alive, you're no longer making ATP. Since you're not making ATP, these myosin heads will never let go again until they just naturally get like destroyed. So if you're trying to move a dead person's arm, you are fighting against all of the myosin heads that are bound to every single thin filament, right? So it's kind of like stuck there like super glue. There's no letting go of that. That is the biochemical basis of what we call rigor mortis in a nutshell. Like everything I talk about, it's probably way more complicated than that, but that's generally what happens. Any questions about our muscles? How muscles are generally moving? If not, we will move. Doesn't make it tougher for the bugs to eat when we are in rigor mortis. No, if you're talking about big bugs, right? They just eat like, it's the same of us as eating like a dead cow, I guess. I mean, we don't really eat the muscle per se, but yeah, it's just protein that their stomachs have to break down. So that's no problem. If you're talking about bacteria, again, it's just proteins that they have to eventually break down. And what will happen, your body will also start to break down itself because we create enzymes that break down other proteins that don't like really require that much ATP all the time. So you'll start the self breakdown, but it shouldn't make us harder to eat. No, not that I can think of, at least. It is pretty metal. I agree. The circle of life and all that. One day we'll be worm food, I guess, depending on where you die. On that cheery note, whoops, I don't want to end the meeting. I want to go to the next slideshow. Okay. Let me go to the right slide. So we just finished talking about muscles. I don't want to present online. What am I doing? Now we're going to talk about the protein basis of the immune system. Specifically, we're going to hone in on antibodies. Like we talked about muscles, we're going to give a big overview of the basics of the immune system works to get just a better idea of or better understanding of how antibodies work and the role that they play. All right. So your immune system, your response of the immune system, your favorite. Well, to be fair, I've never actually taken a class on the immune system, so I'm going to do my best of the overview. So I can talk about antibodies a lot, but the whole system, not my strong suit, but I'll do my best. So when your body reacts to something, we call that something an antigen. So every immune response is triggered by what we call an antigen. Usually these are foreign macromolecules, a protein, a foreign carbohydrate. But if you have an autoimmune disorder, right, the antigen is actually some part of your own body. And what generally happens is that we have B cells. And on the surface of B cells, we have immunoglobulins. And certain B cells can bind certain antigens. And when it binds this antigen and engulfs this whole antigen antibody complex, right? So it kind of eats it, right? And then we have T cells that can stimulate B cells, and these B cells can start spitting out antibodies. These antibodies will start to bind antigens, the same antigen, and we will start recognizing any foreign material. So when you get sick, right? And what the chart is showing down here is kind of like, I know the time scale is not shown probably on your screen, but this is days. So each line is 7 days. So when you first get sick, right, you don't really have antibodies to what's making you sick, because what's making you sick is a foreign invader, some kind of foreign macromolecule. Eventually, you'll create an antibody, or hopefully eventually you'll create an antibody that will be able to attack this antigen. So the amount of antibodies will skyrocket as you keep making more and more and more to attack this antigen, right? You're also creating memory B cells that will just stick around in your bloodstream. And so what's happening here at this little arrow in this picture is that we're getting infected again by the same thing. And since we already have memory B cells floating around that create antibodies for things we've seen before, our production of antibodies for this macromolecule will skyrocket at a very fast rate. So let's say the first time we got affected, we made 100 antibodies. This time, the next time you get infected, you're making, let me just do this math here, 1,000, 10,000. You're making roughly 100,000 antibodies now. So we're really good at fighting the same thing again. So if we've seen it once, we're going to create our memory beta cells so we can, pump out antibodies to catch this antigen. And what we're really going to focus on is we're just going to focus on the little antibody itself, not so much this whole system. We're going to just focus in on the antibodies. That's all we're really interested in when we come to the immune response. All right, so your body has at least 5 different types of antibodies. They're called immunoglobulins. We're going to just focus on immunoglobulin G for our presentation in class, right? And so here, and those different antibodies, they recognize like different things. So you have antibodies for like that are good at recognizing fungi. There's just different antibodies that specialize in recognizing different things. We're going to just look at the most common one, IgG, immunoglobulin G, right? So antibodies generally kind of look like a giant Y, in which you have 2 different fragments. You have a antigen binding site, so that's in this pink. So this pink is going to bind your antigen, and you have this FC region, this constant region. So the antigen binding region, this is variable. So what we're seeing these V's, that means the amino acids change. Each antibody that you make will have a different variable region. The constant region, which is what these C's, they don't change. Every single antibody or IgG that you produce will have the same constant region, but different variable regions. And that all has to do with the complex way that DNAs are produced. The mRNA of DNAs are produced. There's a lot of mixing and matching that goes on, so that every time you make a new antibody, you are making a new variable region. You have different amino acids there to attempt to recognize different antigens. We can roughly split our antibody into two parts. The arms of the Y are called the FAB region or the FAB region, and the tail of the Y is called the FC region. AB stands for antigen binding site. The C region stands for crystallization. FC regions, when you cut them off, they'll make crystals super easily. The antibody itself is made out of two different chains. A light chain, so the light chains are up here, which I just circled, and the heavy chains. The heavy chains are everything else, and you have two of those. So the red there are your two heavy chains, the black are your two light chains. I'm just going to undo that so we can see better. They are all connected by disulfide bonds, right? So your heavy and your light chains and your two heavy chains are bound by disulfide bonds, which are produced by, are made through cysteine residues. So cysteines are holding together your antibodies. And I'm just circling all the disulfide bonds there. Every single B cell in your body will produce a different type of antigen. And the amount of antigen, or sorry, will produce a different antibody. And the amount of antibodies you can make are such a ridiculously huge number, right? It's like, I forget exactly what it is. It's like times 10 to like the 12th or something like that. Just such a large number. But at any given time, we're only making a certain amount of antibodies, right? And that makes a lot of sense that we want to make antibodies that have so much variation, because you never know where you're going to get sick from, right? So we have to get ready for the possibility that we can recognize anything foreign inside of us and make an antibody that will bind that foreign molecule. All right. So that was a lot of information on one slide. But are there any general questions or any questions about our IgG structure here, the amino acids it has, or anything like that? I'll move on here. Okay. So let's just see if we got, how this antibody is laid out. So light chains of antibody are 23 kilodaltons. Heavy chains are 65 kilodaltons. So let's say I ran an antibody through gel filtration. What mass would I see come out, right? What if I put an antibody in through gel filtration? What mass do I think my antibody would be? So what is the mass of the antibody? What mass? If I ran an SDS page with and without 2-mercaptoethanol, what would my gels look like? So for B and C, we're doing SDS page. We're doing one without beta-mercaptoethanol, and we're doing one with beta-mercaptoethanol. So see if you can do those questions. And that's probably where we will, let me just check here really quick. I'm guessing that's where we're gonna call it for right now. Unless we do this really fast. Actually, we're probably gonna go one more slide. Just talking to myself here. So 23 times 2 for both the light chains and the arms, and then plus 65 for one. So there's 2 heavy chains. All right. So if I do gel filtration, your math is more or less correct. it would be the weight of 2 light chains plus 2 heavy chains, right? So each antibody is made out of 2 light chains and 2 heavy chains. Beta mercaptan ethanol. Who can tell me what beta mercaptan ethanol does again? What does BME do? Breaks disulfide bonds. Fantastic. So if you had an antibody and you ran it on an SDS page, you would get one giant band. That would be at roughly, oh, what was my math again? 176 kilodaltons. That 176 is this math, 2 times 23 plus 2 times 65. If you did have beta mercaptan ethanol, all your disulfide bonds would break. And what you would see is you would see 2 bands. You would see one band at 23 kilodaltons, and you'd see another band at 65 kilodaltons, right? Because the light chains are the same weight, the heavy chains are the same weight. So even though we have 2 light chains and 2 heavy chains, we won't see 4 bands. You would see 2 bands because we only have 2 different weights. Questions about that idea, further exploring the structure of our antibody? How do we answer A and B? A is basically what is the weight of an antibody. So I give you the weights of light chain and heavy chain. You just have to know there's 2 light chains and 2 heavy chains and add that together. B is, I kind of modified it. B is do an SDS page without beta mercaptan ethanol and do it with mercaptan ethanol. So B is without BME. So that's why I drew to a gel with that split in half like that. Anything else? If not, we'll go one more slide. We have time for one more. Yeah. Now, when we look at the structure, the actual secondary and tertiary structure of our antibodies, we see that our antibodies have a fold called the immunoglobulin fold. All of our antibodies have this, right? And what it is that we have 3 to 4 anti-parallel beta sheets that are like in a sandwich. And it's hard to see in 2D. What they're talking about, like a sandwich, is that like the blue part is one piece of the bread, the red part is the other piece of the bread, and they're held together by a disulfide bond. So we have I like to think of it like a crab claw almost, where we have this like little sandwich that will go and like bind the things, right? And the beta sheets are kind of don't really change. Where we do see changes in our antibody are the regions called the hypervariable loops. And these loops, anytime you see the word loop, it's almost the same as like IDP. These loops don't have a set structure, right? So hypervariable in that they can be any amino acid, and it changes for antibodies. This is what allows us to bind any antigen in the world, is that these loops will constantly change. Every time we make a new antibody, it'll have a different hypervariable loop. And when we bind our antigen, it is always non-covalent. We do not form covalent bonds. Our binding is only based on hydrophobic interactions, making hydrogen bonds, making ionic interactions. And the binding of antibodies extremely strong. I know these numbers probably don't make much sense to you, but anything that's like 10 to the minus 7 or lower KD means this binds super strong. You only need like nanometers of an antibody to bind your antigen. It has incredibly strong binding. And the cool thing about IgGs, if you remember, it's like a Y, and that's what's being shown here. Your antibodies can bind two things at once with each little crab claw. Other antibodies, like your IgM, so anytime you see Ig, that just means antibody. The big letter tells you what class. So IgMs and IgAs can bind more than two things. They're multivalent. Like we have one type of antibody that can bind 5 things at once, right? And what happens is that when we have, antibodies that can bind 2 things at once, they will bind 2 things at once, like it's being shown here. And foreign substances usually have more than one antigen. Remember, an antigen just means an antibody binds it. So you can have multiple sites where different antigens will bind. And what will happen, or what can happen, is that you'll make different antibodies that recognize different parts of this foreign molecule. That's what's being shown here. So blue, yellow, and green are all different antibodies, and they're all recognizing a different part of this invader. And your antibodies will all glob onto these invaders, and they'll bind 2 invaders at once, and they'll form like a web or a lattice where all these invaders are kind of just like stuck in a giant web. And what will happen is that your body will come and eat these, almost not quite like Pac-Man, but kind of like Pac-Man, where they'll just go and clean up this mess. And so that's one way that we can get over an infection. We just spit out all these antibodies. They will just glob on to our foreign invaders, form this giant lattice, kind of just make it so our invaders can't really go anywhere anymore. They're just kind of like stuck in this web of antibodies and antigens, and then get destroyed. And that is, in a nutshell, how your antibodies help you from not being sick. Questions about that? We'll go over this on Monday, where we'll have questions related to this. But while we're looking at it right now, any questions about that material? All right, if there's not, again, test is on Friday. I will be mostly available on Thursday. I do have a meeting scattered, a couple meetings scattered here on Thursday, but I do not teach on Thursday. So I will be available. So if you have questions about the test, please do shoot me an email. I'll have the test finalized on Thursday. I'll put the equation sheet up on Thursday. Yeah, as always, like I just said, email me. Other than that, I have nothing left for you. I wish you all good luck. Study hard, and I will see most of you on Monday, hopefully. All right, take care, everybody. Good luck.